Welcome, everyone. On behalf of Cambridge Health Tech Institute's Global Web Symposia Series and our sponsor for today's event, Wyatt Technology Corp., I'd like to welcome you to Vaccines Well Characterized, Light Scattering Solutions for Biophysical Characterization in Vaccine R&D. My name is Elizabeth Lamb, and I'll be your host and the moderator for today's event. I'd like to introduce our presenter for today. Dr. Michelle Chen is the Director of Analytical Services at Wyatt Technology Corp., overseeing new application development, sample analysis services at Wyatt's Application Lab, technical sales support, and instrument quality control. She graduated from the Chemical Engineering Department of Yale University, where her doctoral work focused on the development of novel approaches for high-speed and high-efficiency liquid chromatography of biopolymers. Since joining Wyatt Technology in 1996, Dr. Chen has relentlessly pursued new ways of characterizing synthetic and biological polymers by incorporating multi-angle light scattering and dynamic light scattering detection with HPLC and field flow fractionation. So with that, Dr. Chen, I'm going to pass the presenter ball over to you. Welcome.
Thank you very much, Elizabeth. And good morning, everyone. Most of us have taken vaccines at some point in our life. We want the vaccine products to be not only effective, but also safe. To ensure the safety and efficacy of the vaccine product, it is crucial to characterize the vaccine products thoroughly and throughout the entire development and manufacturing process. In today's webinar, I'm going to discuss light scattering and light scattering related tools for biophysical characterization of the vaccine products in research and development. Light scattering tools on their own or used in combination with a separation technique have widely been used in vaccine development, including in developing vaccines to treat and prevent diseases from the Ebola virus. The research group from Scripps Research Institute used the SEC-MALS to characterize a glycoprotein from the surface of the Ebola virus. The glycoprotein is the antigen, and they used SEC-MALS to measure the molecular weight and also the degree of glycosylation of this glycoprotein. Then they used the composition gradient multi-angle light scattering to study the binding between this glycoprotein antigen with different antibody candidates. Vaccine development is a very complicated process because there are so many different compounds that need to be characterized. We will just focus on the characterization of macromolecules and nanoparticles in vaccine development. So first, the antigen. Antigen is the macromolecule that induces the immune response in the body. Antigens can be proteins or polysaccharides. Adjuvant. Adjuvant is the substance that boosts the immune responses to the antigen. Currently, the only FDA-approved adjuvant is the group of aluminum salt, but there are other future adjuvant candidates that are being actively researched. They include liposomes, emulsion particles, virus-like particles, or other viral vectors. Vaccine products. They can be attenuated or kill the viruses. Some of the flu viruses belong to this category. Protein-polysaccharide conjugates and polysaccharides are vaccines against diseases like pneumonia and meningitis, the diseases from bacteria. Virus-like particles are vaccines against the viruses like hepatitis B virus or HPV. Now I will recategorize those different compounds into three major groups of analytes and discuss how light scattering techniques can be used to characterize those analytes. The first group is protein. We can use dynamic light scattering to screen aggregates, also to study the stability of the protein solution. SEC-MALS, size exclusion chromatography with multi-angle light scattering, can be used to measure the absolute molecular weight of the protein or oligomeric state of the protein in solution without assuming the shape of the protein. Composition gradient multi-light scattering, as we mentioned earlier, can be used to study the binding between the antigen and antibody. The second group includes protein-polysaccharide conjugates, glycoproteins, and polysaccharides. They all share the same attribute, that is, they are very polydispersed in molecular weight. So usually SEC is used for the separation, and MALS is used to measure the absolute molecular weight and molecular weight distribution. When MALS combined with two concentration detectors, UV and RI, we can also calculate the composition of the conjugate. When SEC separation is not adequate, either due to column interaction or the polymers are too large for the SEC separation, then FFF can be used as an alternative or sometimes even as a replacement to SEC. The third group of analytes includes different nanoparticles like viruses, VLPs, and liposomes. Dynamic light scattering can be used to measure size distribution with relatively low resolution and in a semi-quantitative fashion. It is also a great tool, especially in the format of the PlateReader, to screen different formulation conditions. For high-resolution size distribution, then field flow fractionation can be used for the separation. MALS can be used to measure the size distribution, to count the particles, and also to reveal shape information for those nanoparticles. CG-MALS can also be used to study the binding involving those nanoparticles, like the VLP with antibody for the affinity and stoichiometry. Now I will give a brief introduction to three major light scattering techniques used for macromolecule and nanoparticle characterization. The first is the multi-angle light scattering, or simply MALS, or static light scattering. In MALS measurement, the scattered intensity from different scattering angle is measured simultaneously. And the scattered intensity at any specific angle is directly proportional to molecular weight, right here in this equation. So by measuring the scattered intensity at multiple angles, we can get the absolute molecular weight regardless of shape of the molecule. If the size is larger than 10 nanometer, we can also measure the loop mean square radius, or radius gyration, RG. When we measure the intensity from two different species at different compositions, then we can measure the binding affinity or the equilibrium dissociation constant. When we measure the intensity at different concentrations, we can obtain the non-specific interaction parameter, second Bureau coefficient, A2 or B22. Dynamic light scattering, on the other hand, can measure the fluctuation of scattered light as a function of time. And from that, we can calculate the translational diffusion coefficient. Then we can obtain the hydrodynamic radius. We can also measure another non-specific interaction parameter called a KD, small k, big sub d. Not to be confused with the other KD, big K, big sub d, the binding affinity. There are two different things. The KD from MALS is for the interaction between molecules. And the KD from dynamic light scattering can be used together with A2 to predict the stability of the protein solution. I will have an example to present later in the webinar. The third light scattering technique is called phase analysis light scattering, PALS. It measures the velocity of a charged species in the electrical field. From that, we can measure directly the electrophoretic mobility, and then we can calculate zeta potential or effective molecular charge. Zeta potential is a good indicator of colloidal stability of nanoparticles. But because of the time constraint, I would not discuss on this topic any further. I will refer people who are interested to learn more about PALS to listen to our archived webinars on our website. Now, both MALS and DLS can be used on their own or in combination with SEC and FFF. When those orthogonal approaches used together, they can give comprehensive and complementary information. Together with other techniques, we can put the pieces together and solve a big puzzle of either a protein solution or particle suspension. I will start with CG-MALS, then go counterclockwise to discuss each of those four different light scattering related tools and their application examples. Composition gradient multi-angle light scattering was first described by Drs. Entry and Minton in two of their analytical biochemistry articles in 2005. It was later commercialized by Wyatt Technology, and the product is called Calypso. In a Calypso experiment, the protein solution or the buffer is filled into one of the three syringe pumps. The flow rates of the syringe pumps are programmed to deliver a mixed solution at the desirable composition and concentration into the light scattering and concentration detectors. After each of such injections, the flow rate is stopped to let the solution to reach the equilibrium inside the flow cells for simultaneous MALS and concentration measurements. Now let's take a look at the results on the right graphs. The top graph is the concentration of two proteins, X and Y, over time at multiple compositions. If there was no interaction, the light scattering signal would just be linear over time or with concentration. When proteins X and Y form the complex, molecular weights increase. So then we will see this characteristic curvature with the maximum in light scattering signal. The extent of the signal is related to the binding affinity or the equilibrium dissociation constant. The location of the maximum is related to the stoichiometry of the complex. So now let's go back to this Ebola antigen antibody finding study. The experiment can be divided into three segments. The first is just the self-association of the antibody. So it just measures the scatter intensity at different antibody concentrations. The middle part is the heteroassociation of the antibody and antigen at 15 different compositions. And the last segment is the self-association of the antigen. The results of antibody and antigen showed constant molecular weights across the concentration range studied. And also their molecular weights are in agreement with their monomer molecular weights. So we can conclude both antigen and antibody do not have self-association and they're in monomeric state. The heteroassociation part, we did see this characteristic curvature with the maximum in light scattering signal. And the maximum occurred at the antigen molar concentration twice of the antibody molar concentration, consistent with the stoichiometry of one antibody, two antigens. And with further analysis, we find the binding affinity to be 90 nanomolar. Comparing to other techniques used to quantify protein interactions, CG-MALS has several advantages. The results from CG-MALS are more intuitive because it's directly associated with molecular weights that are measured from MALS based on first principles. There's no need for sample tagging or immobilization. There's also no solvent restrictions. It can measure not only hetero interactions, but also simultaneous self-association. It can quantify both stoichiometry from about 100 picomolar to millimolar. But CG-MALS does require the complex molecular weight change to be at least 10% upon binding. Also, it requires a minimum amount of 50 to 100 microgram of each sample. Now I will discuss the second light scattering tool, dynamic light scattering, in the standalone mode. Dynamic light scattering measures the rate of light intensity fluctuation, and from that we can measure hydrodynamic radius. Dynamic light scattering can also estimate the different populations, different size populations in the sample. So often it's used for particle size distribution with a relatively low resolution. When we have to screen a large number of conditions, if we use the cuvette-based traditional dynamic light scattering instrument, it will take too much time and it's too labor intensive. So for high throughput screening applications, the high throughput dynamic light scattering instrument is the ideal tool. The high throughput dynamic light scattering instrument from Wyatt Technology is called DynaPro PlateReader. In the PlateReader experiment, the sample solutions are loaded onto the wells of an off-shelf well plate. It can be a 96 or 384 or even 1536 well plate. The loaded plate is then inserted into the PlateReader instrument right here. The laser moves to different wells as programmed for dynamic light scattering measurements. The results can be displayed in many different formats, including the spectral view here. And we can use the color gradient to specify a different size range. So at a glance, we can pick out the wells with the desirable size. For example, we want a certain size, say, indicated by the blue color. And then we can go also into each individual wells to see details. Let's take this blue well, and we can see a nice autocorrelation curve. When the data quality is good, then we can be assured that the size measurement is likely to be accurate. When the autocorrelation curve is not as expected, then maybe we can use the image from the onboard camera to help us troubleshoot. Maybe there was an air bubble or the sample precipitated during the measurement. I'm going to show 3 different application examples with the DynaPro PlateReader in the next 3 slides. High-throughput dynamic light scattering is a great tool to screen different vaccine candidates because it's fast, it's automated, it uses small quantity of samples, it does not have concerns for cross-contamination, and also it provides pretty good characterization ability for size. So in this slide, we show the results from a paper by Professor Finn's group at Scripps. They wanted to study mutations at different sites of a capsid protein for the VLP size and VLP assembly stability. So they have 4 mutant VLPs, and they measured in parallel in the DynaPro PlateReader at an elevated temperature of 60 degrees Celsius over time. So from the time the VLP takes to form aggregates, the aggregation rate or aggregation time, then they can assess which mutation compromise the stability of the VLP the most. In addition to the aggregation rate, we can also use another parameter to assess the formulation stability, the aggregation temperature. Aggregation temperature is the temperature at which the radius rises rapidly. The higher the aggregation temperature, the more stable the formulation is. So in this work published by Professor Middleburg's group at University of Queensland in Australia, they used the DynaPro PlateReader to screen 28 different formulations. And they find the two formulations, both with 0.5% polysorbitol 20, with the additional either 40% sorbitol or 40% sucrose, have the highest aggregation temperature, so indicating those two formulations are most stable. Then they use the FFF-MALS to further confirm the stability of those two formulations in freeze-thaw cycles. In the paper, they also pointed out that it is crucial to actually also screen the formulations in the early stage of the vaccine development. So we know in the early stage vaccine development, the sample quantity is very limited. They only needed 2 micrograms of each of those formulations to study the stability. So that makes the DynaPro PlateReader suitable for even the early stage formulation screening application. The third example is to use the high-throughput dynamic light scattering to measure the propensity of aggregation. And for that, we use the small k big sub d from dynamic light scattering. This KD from dynamic light scattering is measured from the plot of diffusion coefficient versus concentration. So basically, it's from the slope of diffusion coefficient plots versus concentration. So what can we use this KD for? In 2011, almost the same time, the scientists from Amgen and Roche published their work showing very strong correlation between this KD from dynamic light scattering and second Bureau coefficient. We know second Bureau coefficient is from mouse measurements. Usually it takes a lot of sample and it's very tedious to carry out the measurements. KD, on the other hand, is much easier to measure in the high-throughput dynamic light scattering. So because of the strong correlation between KD and A2, we can use KD instead of A2 as an indicator for propensity of aggregation. So in addition to the aggregation rate, aggregation temperature, KD from dynamic light scattering is another good parameter for formulation screening and optimization. Now I will go to the third light scattering tool, that is to use light scattering detectors with size exclusion chromatography. In a more comprehensive system, I will use UV MALS with embedded quells and differential refractive index detector. Here is an example of IgM, two forms, pentamer and hexamer. IgM is actually the first antibody response to an antigen in our body. It is also by far the largest antibody in human circulatory system. In our serum, the predominant form is the pentamer, even though hexamer can exist too, just to a much lesser extent. So the UV traces show the chromatograms of pentamer in pink and hexamer in blue. We can see those two main forms eluted at very similar time. They each also has some aggregates. Light scattering was able to measure the correct molecular weights for each of the form and their aggregates. So even though they may elute at very similar times, but we can see their molecular weights are different. So this shows the importance of using the MALS after size exclusion chromatography in order to measure the correct molecular weight. IgM is actually heavily glycosylated comparing to IgG. The degree of glycosylation, the average degree of glycosylation is about 10%. So we can use MALS together with two concentration detectors, UV and RI, to not only measure the molecular weights, but also to tease out the contribution from each component. So we can measure here the total molecular weight of this glycosylated IgM and also the molecular weight from the protein component as well as the molecular weight from the carbohydrate component. How can we do that? We actually have an archived webinar on protein conjugate analysis if you want more details. But just very briefly, I'm going to say that that's actually the composition is from the mass conservation from the two concentration detector. UV is responsive to only the protein portion, and RI is proportional to both protein and carbohydrate. At each data slice, the concentration should conserve. So from that, we can tease out the composition. Now, if we add dynamic light scattering to SEC-MALS setup, then we can also get information on the shape. So here are some examples of interleukins. Interleukins are actually very important for the proper function of our immune system. Some interleukins are also used as adjuvant. So we have one protein in red. It's interleukin-4 trap protein. It's a relatively large protein, 240 kilodalton. And the blue is the complex of two interleukin-4 proteins bound to the trap protein with a measured molecular weight of 275 kilodalton. Both proteins show a very homogeneous peak because the molecular weight across the peak is flat, indicating there's only one protein in the peak. However, what's strange or interesting here is the complex. The complex has higher molecular weight, yet it eluted later. We know in size exclusion chromatography, the large molecule is supposed to elute earlier. The late elution is usually caused by column interaction or by a more compact structure. So we can use the online dynamic light scattering to help us to find out which one is the true cause. The online dynamic light scattering measured 7.7 nanometer for the trap protein. And the complex, even though it has a higher molecular weight, the hydrodynamic radius is only 6.5 nanometer. So we know in size exclusion chromatography, the elution order is based on hydrodynamic radius, not directly by molecular weight. And so that explains a more compact structure of the complex is the cause for the late elution. Other analysis also confirms that this complex structure can collapse when the complex is formed. MALS intensity is proportional to both concentration and molecular weight. Most of the vaccine components are pretty large, so MALS sensitivity usually is excellent. But when you work with small particles or proteins, and especially with low sample amounts, like in the case of early vaccine development, then it is very important to make sure your MALS sensitivity is adequate. We have example of HIV capsid protein here. with molecular weight around 10 kilodalton. We only needed 10 microgram injection to get a very nice data quality for accurate molecular weight measurements. So I often use this formula to estimate the minimum concentration needed at the MALS flow cell. In a more practical term, probably we want to know what is the minimum SEC load when we use the detector with SEC separation. So you can use this formula to estimate the minimum load on the traditional column dimension, 7.8 millimeter in the diameter and 30 centimeter in length for signal-to-noise ratio about 5. It is not surprising both formulas contain the molecular weight term. That's because the light scattering intensity is proportional not only to concentration, but also to molecular weight. So let's take a moment to look at this minimum load. If our protein is 10 kD, like this HIV capsid protein, then the minimum load is only 1 microgram. When our protein is 10 times larger, 100 kilodalton, then the minimum load is only 0.1 microgram. If we're using a narrow bore column, then the amount required is only a third. And the UHPLC column would reduce the minimum load even further. The last light scattering related tool I'm going to discuss today is the field flow fractionation together with the light scattering detectors. The modern field flow fractionation, usually the channel width tapers down towards the outlet, so it's known as asymmetric FFF. In FFF, usually a field is applied perpendicular to the channel. Depending on the type of field, we can have different types of FFF techniques. The most versatile field is actually a flow of the mobile phase perpendicular to the channel, known as the cross flow. The action of the field, the cross flow, will push the samples against the bottom wall. But samples would diffuse back. Small ones diffuse faster and more away from the wall. The big particles diffuse slower, they're closer to the wall. Along the channel, we have another flow. It's called channel flow. Channel flow will have the parabolic or laminar flow profile, which means we have a flow velocity gradient perpendicular in the vertical direction of the channel. The small molecules or particles, they're more away from the wall into the higher flow velocity region. So the small ones elute first. Large ones scattered more into the low flow velocity region. They elute later. So the elution order in field flow fractionation is just the opposite as in size exclusion chromatography. Here is a picture of the FFF setup, and we can see they share many common components as the SEC setup, the same pump, auto sampler, and the suite of detectors. So actually, if you have a FFF system, then you can also do SEC. So you can do both SEC or FFF separation in the same system. The asymmetric flow FFF, or simply AF4, is a non-destructive technique for particle separation or for separating very large polymers. It has a very wide size range from 1 nanometer to several microns. It can be automated with the auto sampler just like the typical HPLC system. It can separate particle-containing sample that can foul the column easily. As I mentioned, if the molecules are too large for the SEC separation, then FFF is a good replacement. It can use together with the different detectors, including MALS and DLS, for online absolute characterization. Now I will have some examples to share. Particle size distribution is often first attempted by dynamic light scattering because dynamic light scattering is more readily available and does not require much method development. However, if high resolution size distribution is desirable, if you want to see small differences between samples, then FFF is probably needed. So we have an example of adenovirus here. We made 2 injections with the FFF separation. We can see very good reproducibility. The majority of the particles in this adenovirus sample has a radius of about 45 nanometer, consistent with 1 adenovirus particle. But we also detected a small peak at the tail region of the main peak with a radius of 75 nanometer. And this is consistent with 2 adenoviruses bound together. This small amount of large aggregate will not have been detected by the dynamic light scattering. By adding MALS detector to FFF, not only allows us to measure the size and size distribution, it can also count the particles. In this paper published by the scientists at MedImmune, they used both FFF-MALS and AFM-TM to count the particles of the adenovirus. And they find the results are in great agreement. 2.8 times 10 to the 10th from FFF-MALS versus 2.9 times 10 to the 10th. So validating FFF-MALS for particle counting application. When we add dynamic light scattering to FFF-MALS, then we can measure the shape of the particle. So MALS will allow us to measure the mass average, the radius gyration, or G, and dynamic light scattering allows us to measure the hydrodynamic radius, or H. The ratio of RG and RH can help us measure the shape of the particles. So the first peak here is in dark blue, has a RG-RH ratio about 7.77, consistent with a solid sphere structure. The second one in red has a broader peak and also has a RG-RH range from 1.2 to 1.5. It's more like a rod. So by measuring both RG from MALS and RH from dynamic light scattering, then we can get a shape of the particle. Now that I have discussed all those 4 light scattering tools, I'm going to present some case studies. The first case study is with protein-polysaccharide conjugate. Those are the vaccines against the diseases from bacteria. And they're usually very polydispersed in molecular weight, so a separation technique is needed. SEC usually is the first technique to use because it's more available for most labs. But recently, we used both SEC and FFF to measure some high molecular weight protein-polysaccharide conjugates. And I'm going to share those results with you today to compare SEC versus FFF for those conjugate separation, and also to discuss some potential advantages of using field flow fractionation for the separation. The first protein-polysaccharide conjugate example has very wide molecular weight distribution, varying from 1 million to 100 million. The results from SEC is in red, showed a molecular weight decrease with elution volume initially, then curve up at a tail region. This is likely caused by an effect called anchoring effect, and I will discuss a little bit more in the next slide. But this anchoring effect is not seen from the results from FFF. We can see the molecular weight from FFF monotonically increasing with elution volume. the results from 2 injections also are very reproducible. We also find the molecular weights from FFF are higher than the molecular weights from SEC. The weight average molecular weight from FFF is about 14 million Dalton, and from SEC is about 10 million Dalton. So from those results shown in this slide, we can see the 2 advantages of FFF separation. One is that it has less shearing degradation, and it has no anchoring effect. So what is this anchoring effect? The anchoring effect is actually one of the 2 major non-ideal SEC effects, the other being column interaction. Usually it happens to large polymers with multiple chains, like in the case of a branched polymer. So the one chain or multiple chains can be anchored onto the pores of the SEC packing, just like a boat anchored to the sand. The anchoring effect will cause the high molecular weight fraction to have a delayed elution, or will co-elute with the low molecular weight fraction. And this is more prominent in the tail region. That's because light scattering measures weight average molecular weight at each eluting slice. And at a tail part where the low molecular weights elute and the co-elution of the large molecule will make the molecular weight to curve up versus elution volume. So when there's a non-ideal SEC effect in the separation, the weight average molecular weight usually is not affected, but number average molecular weight will be overestimated and polydispersity underestimated. It will also affect the measurement of the conformation. And the conformation is measured from the conformation plot. That's a plot of radius versus molecular weight. So when we have this anchoring effect, the data from SEC usually will show this characteristic curvature, or the Nike sign. But the results from FFF showed linear trend. So we can then measure the slope of this conformation plot correctly, and so we can measure the conformation. So from this, we can see that the results from SEC, when there is non-ideal SEC effect, we would not be able to measure the conformation accurately, but we can with the FFF data. The second protein-polysaccharide example also showed curvature in SEC data, but the molecular weights from FFF still monotonically increase with elution time or volume. But also we can detect an aggregate peak from the FFF separation, and this was not seen from the SEC separation. So this shows FFF can detect high molecular weight fraction or aggregates that cannot be detected by the SEC column. Likely they're being removed by the SEC column, either by the column fritz or by the packing itself. Protein conjugates, not only they're polydispersed in molecular weight, often they're polydispersed in composition as well. So it is desirable to measure the composition across the peak. So I showed a couple examples before to use UV, MALS, and RI to measure the composition. So I will have another example to show here too. That is the same protein but expressed from different cell lines. So the molecular weight from the protein portion for both samples overlapped because they're the same protein. But the one expressed from mammalian cell has higher content of carbohydrate, and the degree of glycosylation is about 15%. The molecular weight from insect cell has lower content of the carbohydrate with a degree of glycosylation of only 10%. So by using the light scattering detector with UV and RI, we can reveal that those two samples have the same protein but different contents of carbohydrates. However, when we use protein conjugate analysis to measure the composition of those high molecular weight protein conjugates, we encountered a challenge. On the left graph, we have fractograms, the traces from FFF separation from different detectors. The red is from light scattering, blue is from differential refract index detector, and the green is from UV. When the molecular weight is over several million Dalton, UV trace will often have an artifact that's from scattering, actually. So this will impact the left half of the peak for a SEC separation. That's where the high molecular weight fraction eludes. For FFF peak, usually it will affect the right half of the peak. So when we use protein conjugate analysis, we will find the carbohydrate content will drop to 0 for the second half of the peak. That's because the UV, due to the scattering artifact, that UV signal is overestimated, so the protein content is also overestimated. So if we still want to do protein conjugate analysis, we have to remove the scattering contribution in the UV signal first. And we're currently working on the correction in our Astra software. So I'm going to summarize the advantages of the FFF separation comparing to SEC for those high molecular weight protein-polysaccharide conjugate characterization. FFF has less shearing degradation. It does not have the anchoring effect because there's no column packing. So then it can measure more accurate molecular weight distribution, polydispersity, which is defined as the weight average to number average molecular weight, and conformation. It is also allow us to measure the ultra-high molecular weight fraction that can be removed from the SEC column. I will caution, because of the scattering artifact in the UV signal, we will have the same problem whether the separation is performed by the SEC or by the FFF. But hopefully we will have a correction in our software soon, and I will share that when we have that correction. The second case study is with liposomes and emulsion nanoparticles. Those are candidates for adjuvant and also can be used for vaccine carriers. So we have two liposomes here. One is empty, the other is filled. And first, we will always use dynamic light scattering to assess the particle size distribution. So on the left are the autocorrelation curves from those two liposomes. And the right are the size distributions. And we can see both results showed very similar graphs in the differences within the statistic error. So from dynamic light scattering, they seem very similar. Now we also use the field for fractionation combined with online dynamic light scattering and multi-angle light scattering to characterize those two liposomes. The RH versus elution time for those two liposomes overlap, suggesting the FFF condition is quite optimal because the separation is based on the hydrodynamic radius as you would want from a FFF separation. However, when we plot RG from MALS, we found at each eluting time, the RG from empty liposome is larger than RG from filled liposome. Why is that? Well, that's because RG measures the mass average the radius. So the internal mass will make the RG smaller comparing to the radius of the sphere, which should be very similar to the hydrodynamic radius. So if we calculate Rg to Rh ratio, we will find for solid sphere, the ratio is around 0.77. For a hollow sphere or spherical shell, because most of the mass is on the surface, those pictures are not drawn to scale. So in the case, extreme case where the shell is much, much thinner comparing to the radius of the sphere, then Rg, Rh would just be 1. So that explains that at each eluting slice, we can have the same Rh values for both empty and filled liposomes, but the empty liposome will have larger Rg value than the filled liposome. So when we plot Rg versus Rh, we can also get the Rg-Rh ratio from the slope. So for the empty liposome, the ratio is around 1.0, consistent with the spherical shell structure. For the filled liposome, we found Rg-Rh ratio about 0.77. The numbers, the actual numbers are displayed here. And 0.77 is consistent with the solid sphere structure. So by measuring Rg-Rh from a well-separated particles from field flow fractionation, we can get structural information. Now, MALS not only will help us to measure the size, Rg size, and size distribution, it can also count the particles. And when we integrate the entire peak, then we can calculate the total number of particles in the sample. In addition to the papers published by the scientists at MedImmune, a group at CDC in 2013 also published using FFF together with MALS to quantify the particle numbers in influenza virus particles. And the reference is shown at the bottom of the slide. The third case study is with virus-like particles. In the interest of time, I would not show more examples, but rather I just want to point out high-throughput dynamic light scattering, like the PlateReader instrument, is ideal to study formulations and to also perform accelerated stability tests. SEC or FFF, together with the light scattering detectors, can be used to measure the size distribution with high resolution, to count the particle numbers, and also to reveal structural information. For a more comprehensive discussion on VLPs, you may listen to an archived webinar that was just given a couple months ago. Now I will recap the light scattering tools we discussed just now. So usually we will use SEC or FFF for the separation for those vaccine compounds, and we will add the MALS and DLS for absolute molecular weight and size distribution. For proteins, we can use MALS with UV and RI to study the composition. With the addition of dynamic light scattering, we can get information for the conformation. For particles, we can get the particle shape. We can also use the intensity from MALS for number density of particle counting. We can use high-throughput dynamic light scattering to measure the size distribution. We can also use it to screen and optimize different vaccine candidates and different formulations. CG-MALS is to study the binding affinity between antigen antibody, VLP antibody for both stoichiometry and the binding constant. Hope you will add those light scattering tools to your biophysical characterization toolbox here in your future vaccine development in order to make the vaccine products more effective and safer. I would encourage everyone to visit our website for more theory on those different light scattering techniques, to download application notes, also to search our bibliography for peer-reviewed articles, also to listen to those archived webinars on the topics we only had time to touch on, like PALS, VLPs, FFF, protein conjugate analysis. And last but not least, I would like to thank everyone for your attention.
Thank you very much, Dr. Chen. I do want to thank so much the folks from Wyatt. I'd like to thank you, Dr. Chen, for a wonderful presentation. As a matter of fact, someone just chatted in. Great talk with multiple exclamation points. So thank you so much. And most of all, thank you to all of you who came and spent an hour with us. Hopefully you got some information that will make your job a bit easier. I look forward to seeing you at future CHI Global Web Symposia. Thank you, and have a great day. Bye-bye.
Bye-bye, everyone. Thank you again.